In the interphase, the cell develops and grows into a mature functioning cell, while also duplicating DNA in the nucleus and preparing for cell division. Interphase is divided into three stages called G1, S, and G2, or gap, synthesis, and gap 2. At this point, the nucleus is still intact. In G1, a cell grows, carries out normal cell functions, and prepares to replicate DNA. At this point, some cells exit and do not divide again. In S phase, the cells copy DNA in order to prepare for cell division. And in G2, the cell gets ready for its nucleus to divide. The cell also takes inventory and makes sure it is fully prepared to continue to the next step of mitosis. In prophase, the cell's chromatin condenses to form chromosomes that are shown as X's in these pictures. Each half of these X's are sister chromatids, which are structures used to contain identical copies of DNA. A centromere ensures that a copy of replicated DNA will become a part of the daughter cells at the end of mitosis. The centromeres are the small circle dots in the center of the chromosomes, where the sister chromatids are attached. In this phase, the nuclear membrane and nucleus disappear and the spindle apparatus begins to form. The next phase is metaphase. The sister chromatids line up on the equator of the cell, as shown in the pictures above. They do this because they are pulled along the spindle apparatus by motor proteins. Spindle fibers are now attached to each chromosome's centromere.
In this phase, the separation of replicated chromosomes occurs because the chromatids are pulling away from each other. In anaphase, microtubules of the spindle fibers move the homologous chromosomes away from each other. The sister chromatids split into two identical chromosomes. At the end of this phase, the microtubules move the chromosomes towards the poles of the cell, along with the help of the motor proteins. And as you can see in all these pictures, they are pulling away from each other. Telophase is the final stage of mitosis. The chromosomes arrive at the poles of the cell and begin to settle or decondense. In this phase, two new nuclei are formed, each with a complete set of chromosomes, also known as DNA. As you can see on the right-hand picture, two new nuclear membranes start to form and the nucleolus reappears. Lastly, parts of the cytoskeleton are built by recycled structures used for mitosis by the cell. At the end of mitosis, the cell's cytoplasm divides and the cell begins to divide as well. This is known as cytokinesis. This is resulting in two cells with identical nuclei. Cytokinesis occurs in animal cells and is accomplished by using microfilaments to constrict and pinch the cytoplasm. Unlike animal cells, plant cells have a stiff cell wall. Instead of pinching in half, a new structure called a cell plate forms between the two daughter cells. The cell walls then form around the cell plate. When this is finished, there are two genetically identical cells. As you can see in the bottom right-hand picture, this is what happens. The two cells have divided into two.